Hello everyone, my name is Joaquina Mascarenhas and I'm a manager in the Upstream PD group at Thermo Fisher Scientific. Thanks for joining and allowing me the time to talk to you today. In this presentation, I will speak to how Thermo Fisher Scientific, and specifically the Pharma Services Group, is leveraging innovative technologies to serve the patient population through accelerating biologics development. To begin, I'm going to give a quick introduction to the Pharma Services Group, and then I will get into a little bit about the things we are observing around overall industry trends. I will then describe how Thermo Fisher Scientific, specifically Pharma Services, is leveraging innovative technologies to respond to these industry trends. And lastly, I will briefly touch on some other productivity solutions that we are looking to implement as an organization to better serve the patient population. Today, Thermo Fisher operates the world's largest pharma services business, offering drug substance, drug development, clinical trial services, drug product, and viral vector development and manufacturing solutions to companies of all sizes. Thermo Fisher Scientific is expanding its footprint in Europe and in North America for sterile drug product development and commercial manufacturing of critical medicines, therapies, and vaccines, with the addition of 13 development and commercial production lines to support a range of capabilities, including live virus, aseptic liquid, and lyophilized vial filling. In addition to recent expansions in Europe, Ferentino, Monza, and Swindon, we recently announced significant projects in the Asia Pacific, including a new sterile manufacturing facility in Singapore and a new integrated biologics and sterile drug development and manufacturing site in Hangzhou, China. A key differentiator for us is our ability to support our customers end-to-end, starting at early drug development and through the entire product life cycle. From molecule to medicine, we have the capacity, the capability, and the expertise to support drug developers every step of the way. Even with our current expertise and capabilities, as a CDMO, it is imperative that we keep a close eye on industry trends and continuously evolve in response to those trends. A major trend in the industry is the increasing complexity of the molecules that make up the therapeutic pipeline. The proportion of products sold worldwide that are specialty products has increased twofold over the last 10 years. Overall, we are seeing the transition from small molecules to biologics, biologics being inherently more complex. And not only are the modalities becoming more complex, but the patient populations for each therapies are shrinking and becoming more specialized. Speed is more important than ever as patients are waiting for medicines that treat rare conditions. With the support of the regulatory agencies, one can see a substantial increase from 2015 to 2019 in the percentage of drugs receiving some type of specialty designation. So what does that mean? It means we get to have some more exciting and interesting problems that we need to solve. And even with the increasing complexity, we are being asked to do it faster. So what are some of the things that we as an organization are doing to help address that? A perfect example of this is our accelerated cell line development workflows, enabling us to deliver from gene to the clinic at world-class speed. Cell line development is one of the very first steps in the development of a biologic molecule, in which host cells, in this case mammalian cells, such as CHO, are transfected with DNA containing the gene encoding the molecule of interest. At Thermo Fisher Scientific, we have heavily leveraged the use of highly automated and miniaturized technologies, such as the Berkeley Lights beacon and the Sartorius Amber bioreactor systems, along with high titer and robust host cell lines, to dramatically shorten the cell line development timeline. Here we see Thermo Fisher's cell line development workflow using the beacon and amber systems to get from initial transfection through screening of approximately 7,000 clones in the beacon to research cell banks of top 4 clones selected based on performance in the amber system in approximately 14 weeks. In the next few slides, I will provide more details regarding the adoption of some of these exciting and innovative technologies into our platform and how they have enabled us at Pharma Services to essentially rethink how cell line development is done. The foundation of any cell line development process is to start with an optimum host cell line vector combination. As a CDMO, we need to understand what the customers are looking for in the cell line development process to produce their therapeutic molecule. Things like high titer, short timelines, well-documented regulatory-friendly host cell lines, as well as low licensing costs are what is definitely driving the business. With these goals in mind, we have worked with our colleagues in bioproduction who developed new Thermo Fisher host cell lines to develop a platform. So high titers, we went about 3 grams per liter. We wanted to have a timeline from transfection to top clone between 4 and 5 months. We wanted to ensure that GMP bank cells are available in AOF CD media and that there are no milestones or royalty payments. And the result of this collaboration is the Freedom XPQS system. In the Freedom XPQS system, Thermo Fisher Sciences paired a very robust CHO-S parental cell line with a new expression vector system to enhance the expression of proteins of interest. And while these cells are in final development and will be available on the market soon, we are actively pursuing integration of this cell line into existing Pharma Services Group cell line development offering. Thermo Fisher was one of the first CDMOs to be able to offer cell line development utilizing the Berkeley Lights beacon system as a service to customers. At Thermo Fisher, we are very excited about this technology that has allowed us to really reduce our critical path timelines, streamline our process, and to rethink how CLD is done. The beacon basically performs single cell cloning on a chip, using light to move single individual cells into pens on the chip, holding only about nanoliter of media in each well. The system can also visualize growth of the cells and antibody production at nanoliter levels. This system gives us the ability to evaluate approximately 7,000 individual clones at once, with assurance of monoclonality, allowing for faster cell line development timelines, and also giving us the screening power that we need to identify the highest producers in a population of transfected cells. So with its integrated end-to-end automated workflow for rapid cloning and selection in mammalian cells, incorporating the beacon has had a real impact on our cell line development workflows. In the next few slides, I will dive into the various aspects of our workflow that the beacon has impacted. I will describe how the beacon has enabled a very fast, accelerated workflow, allowing us to shorten the timeline to clinic, as well as increase project capacity and get more molecules to the clinic faster. We have found that the beacon allows us to select the best clones by increasing the probability of finding those rare, high-producing, robust cell lines that would move to larger scales and manufacturing processes. All of this is combined with the ability to support regulatory submissions with validated monoclonality assurance. As described previously, one of the major impacts of the Beacon platform is on the timelines for single-cell cloning and clone selection. Compared to more traditional methods such as limiting dilution cloning, Beacon-assisted cloning is much higher throughput, as well as allows for on-chip productivity assays in combination with the single-cell cloning step, bringing down the timeline from 4 to 6 weeks for a more manual process to less than a week for the Beacon process. Another major contributor to shortening of timelines is the enabling of single-cell cloning from bulk stable pools, eliminating the approximately 8 to 10 weeks needed for mini-pool generation and tighter screens before proceeding to single-cell cloning. As shown in the histograms on the right, our experience has shown that bulk selected stable pools are very heterogeneous with a large proportion of non or low expressing cells. The Beacon platform allows us to screen thousands of clones across different pools, allowing us to only export and scale up the top producers from each stable pool. Now, the Beacon platform is a powerful tool for screening and taking forward only the highest producing cells. However, in order to get the best clones out of a cell line development process with the highest titers, increasing the heterogeneity of the cells loaded on the Beacon system is very important. At Thermo Fisher Scientific, we have developed workflows for increasing the starting transfected cell culture population diversity by loading multiple pools onto a single chip, allowing us to single cell clone from up to 12 stable pools per program. Moreover, loading multiple pools on a single chip allows for a direct comparison of the productivity of clones from those pools in a way that is not possible when only loading a single pool per chip. For example, we can load 3 pools onto one chip as is shown here. These pools need to be loaded all on the same day and then assayed together on the same day. When analyzing the productivity assay results, we can sort all of the clones on the chip by score or RQP to visualize the best clones. As we scroll through the top clones, we can see which pool each clone came from and see how the 3 pools are represented among the top clones. If only 1 or 2 of the pools make up the top clones, we can then filter the results to only see the clones from the absent pool and see how they compare or see how much worse they are. The filter and sorting features in the Berkeley Lights Assay Analyzer software allows us to look at these pools any way you'd like, one at a time, all together, or by field of view. This feature of the Beacon has been very useful for us when working with projects where we are comparing many pools, and we generally load 2 or 3 pools per chip. In order to ensure the selection of the best clones for predictable performance at scale and to really fast track the back end of the CLD process, we have integrated the Beacon system with the high throughput AMBER system for clone evaluation after scale up and select the top clones for future benchtop bioreactor evaluation. This integration not only allows us a very aggressive timeline and rapid narrowing down of the number of clones taken forward, it also gives us a huge pool of data that we can begin to mine to understand the cell lines better and make better cell line selections. This aggressive timeline and narrowing down of clones selected to move forward to the next stage of evaluation is possible because the Beacon gives us a much higher accuracy and confidence in our ranking for the eventual productivity and cell growth that we would see in our small-scale bioreactors. Shown here on this slide are the correlations between the Beacon score and the fed batch performance for the top clones that were evaluated in the AMBER mini bioreactor systems. It is important to remember here that the population represents a fraction of the thousands of clones that were screened on the Beacon, with only the top 48 to 96 clones exported from the Beacon based on the growth, monoclonality, and spotlight productivity assay. As has been widely reported, the Beacon platform workflows allows for the selection of clones with inherent per cell productivity characteristics and a high correlation with eventual fed batch specific productivity. Along with the ability to screen thousands of clones in a single workflow, the Beacon also has had an impact on our overall productivity. The high on-chip cloning efficiency and the optimized export and scale-up conditions allow us to progress more molecules to the clinic in a more efficient manner with significantly reduced resources and shorter timelines. For regulatory submissions, OptoCLD provides visual records documented from before cloning into the pen with the empty nanopens through the final assay and export of clones. In process monitoring of every pen and quality control of every pen provides superior end-to-end monoclonality assurance when compared to microtiter-based cloning methods. At Thermo Fisher Scientific, we have validated our workflows to achieve greater than 99.64% monoclonality assurance. In addition to the technologies that have been described here, we continue to add productivity solutions, including new analytical workflows such as the introduction of multi-attribute method, a workflow that uses a low artifact peptide map for identification and quantitation of post-translational modifications on the amino acid level that provides for orthogonal glycan and charge variant data, as well as information not directly monitored with other assays, such as oxidation, O-linked glycans, and the presence of low-level impurities. Another productivity solution that is showcased here is our new high-performer dyno-drive single-use bioreactor platform that has been redesigned for higher performance through greater power, better mixing, and mass transfer. At Thermo Fisher, we continuously evolve and reinvent our workflows to better serve the patient population. Thank you so much for your time, and I look forward to your questions and would be happy to provide further information on the topics presented today. At the same time, I would like to take a moment to acknowledge some of the Thermo Fisher Scientific team that has been part of this journey and has contributed to the work that has been shared today. Thanks, everyone.